I start with the class, there are some announcement to be done. I came to know that you have your semester exam on the month of March. Okay, so there are a lot of time. There are lots of time still. So and but 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 in case of your cell biology syllabus, I think today we will be doing the cell cycle and its control and regulation, and tomorrow there will be another class where I will be doing the cancer part. So that mostly concludes your theory part of the cell biology. But as there is still a lot of time there, from January onwards, I will be taking revision classes, because still if you have your class, you have your semester exam in March. So I think it's better if we start revising from the month of January. And December, from next week I won't be taking your classes till 26th of December. Your next class will again be on 27th December. Tomorrow is the last class, then again on 27th December. And you have to present your presentation on January, not on December. You will present your presentation on the month of the January. That will be considered as one of the assignments. So, and your practical classes will be starting on 27th December. The first class after the break. On that day, I will be taking your practical classes. In the meantime, during this break, please go through all the topics of your cell biology that are already done, so that it will be easier for you when the revision starts from the month of January. Maybe one or two topics, theory topics, will be left with. Maybe that cell precipitation, cell fractionation of the cell, that we will be doing on the month of the March, a month of the January, while doing the revision. Okay, so tomorrow I think we can be able to finish the theory, mostly the theory part of the cell biology syllabus. And I don't know, because yesterday was the last day for new admission, direct admission. Still there, I came to know that there are still 2 students yet to join. I don't know when will they join, or when will they attend the class, but I think they will get the opportunity of the revision class. Can't be helped, because the syllabus I've already done once. So for that reason, I think, according to your assignments, I don't know when your first assignment, when the date of the submission of the first assignment will be announced. So it's better, before I go to the break, I will provide you the assignment on next week. That is on Monday. I will be uploading your assignments on the Google Classroom. You will do that assignment, and I don't know when the marks, when the date for the marks for entry of the marks will be announced. Please একটু মাইকটা অফ করো।
30 টাকা পেটে দিন। তাই নিন 30 টাকা পেটে দাও।
মাইকটা অফ করো কৃষ্ণকলি।
বোন খাবে, আমি খেতে পারবো না।
So this is all about your cell biology class. Or if you have any problem, if you have any query, I am sorry to say that I can't solve your query during that period, during that break period. You just note down. You just
ম্যাম এক্সিটার
কৃষ্ণকলি শুনতেই পাচ্ছে না। কৃষ্ণকলি
কটা দেবো?
রাস্তায় আছে,
Maybe
শুনতে
she is
পাচ্ছে না।
muted.
Okay, yes, she. Let me check if I can mute her.
Ma'am, you
কি
can
হলো?
mute.
Mute
কেটে গেল
her.
নাকি না?
So, if you have any query regarding your topics about your Selvaji, please note down that and ask me in the month of January, or when the class after the break will start. That is on December 27th, because I can't take that any query, any phone calls during that time of my examination, semester examination. Okay, so please bear with that. Please keep that in mind. I'm requesting you. I can't do that during that break. Okay, so now today let's start with our topic, that is cell cycle and its regulation. So can anyone tell me what is cell cycle? You have, I think you have already know about this topic. So anyone from your class, can you tell me what is cell cycle?
Ma'am, may I?
Obviously you can.
Say, cell cycle is the properly cyclical process of some cellular reactions to develop to develop a cell. It goes in many stages like Z0, next
Hmm.
S, and next G1, G2.
Yes. What is the G0 called?
I think zero state,
Zero state, na? It's called a resting state
resting state. Sorry.
or the interface state. Yes. Yes. So it's okay. The whatever you have said, it's okay. So we can say that cell cycle is a series of events that takes place in a cell as it grows and divides. That is very important. As the cell grows and divides, the cell cycle is a series of uh, events that are taking place within the cell. And after that, uh, then the cell, after division occurs, after cell growing and the repeat and the phases are going, division phases are going on, then there is some phase which we call which we call it as an interface. What happens in that interface? The usually the cell leaves any of the phase and uh, go to a resting state. Resting state means it is the state when the cell completely stops its division. Okay, and it is the time also in this interface or G0 phase that the cell can revive or prepare itself for the next cellular events to occur. So we can also say this interface state as preparatory stage also. Then what happens after it leaves then the cell again leaves that interface state, and then the cell undergoes mitosis, and then after this mitosis a complete division of the cell occurs. And ultimately what happens after complete division of cell? It forms two daughter cells, and these daughter cells again then resume their own cell cycle stages or steps. And the daughter cell also enters their as they also enters their own interface, and then again along with the interface it also begin a new round of cell cycle. And the duration of this cell cycle events mostly is this duration varies from species to species. In human cell or in our human body's cell, the total cycle time is mostly, we take it as 24 hours. Total cell cycle time means total all the events that are occurring in the cell cycle starting from G1 to M phase, and the particular time taking on each of these events on each of these cell cycle phases are: G1 is taking mostly 11 hours. S phase, that is the synthetic or synthesis phase, mostly takes 8 hours. G2 phase, that takes 4 hours. And ultimately the division phase, that is the mitotic phase, that takes only 1 hour. Okay, so we can say that after, so this cell cycle is going on in a continuous form, or it is the continuous process that is occurring inside our human body or in any organism. But there are some control or checkpoints present in the cell cycle. Why these checkpoints or control or regulation points has to be present in the cell cycle? Because these cell cycle checkpoints are very important. It ensures the cell for its further progression, for the cell to divide, whether the cell will enter to the next phase or next stage, or the cell will go to the G0 phase or resting phase. It depends completely on the cell cycle checkpoints. How the cell, what is the health of the cell? Is the cell is capable of further division? Is the cell is capable of going to further stages of cell division? So from that perspective, the cell cycle checkpoints are very important. So we can define the cell cycle checkpoints are the control mechanisms in eukaryotic cell cycle which ensure its proper progression. And what happens, it actually, the conditions of the cells are assessed during the cell cycle checkpoints, and uh, and along with the condition of the cells that are assessed, the progression through the various phases of cell cycle occur only when favorable conditions are met. Okay, there are many other conditions that are to be met to find whether the cell is suitable for the next phase or not. Favorable conditions means whether the cell is whether there are energy molecules are there, whether cofactors are needed, where any proteins are needed. That are the favorable conditions that are also to be met along with the cell cycle checkpoints. Okay, so we know that there are mostly major three cell cycle checkpoints. What are the major three cell cycle checkpoints known as? One is First one is known as G1 checkpoint, and this G1 checkpoint is called the start or the restriction checkpoint, or it can also be known as measured checkpoint. Then the second one happens at G2, in between G2 and M phase checkpoint, which is also known as G2M checkpoint. So, and the third one is known as the metaphase to anaphase transition. This is also important for mostly the cell division, for cell division mostly. And this metaphase to anaphase transition, this checkpoint is also known as spindle checkpoint. So the progression through these checkpoints is mostly largely determined by activation of some protein molecules or protein and enzyme like molecules, which is known as cyclin dependent kinases. And this cyclin dependent kinases, by regulate these are mostly regulatory protein subunits. These are also called only cyclins. And and there are many lots of different forms of cyclins are found during this whole cell cycle phase or cell cycle event. And different forms of which And different forms of the cyclins are produced at each stage of the cell cycle to control the specific events that occur. So we see that this cyclin play a very important role in the control of the cell cycle. So in a whole we can say that cell cycle control or cell cycle checkpoint is that transitions that occur or that even that takes place between different phases of the cycle and are regulated. That's why the name checkpoint. It is the point, or it is the position within the cell cycle where where it need to be, where the conditions of the cells are need to be checked. That is why the name checkpoints. So we can say that checkpoint is a mechanism that can halt the progression through the cycle until a critical process such as DNA synthesis is completed, or until damaged DNA is repaired. So we can clearly understand how important are these cell cycle checkpoints. So coming to the description of the each type of checkpoints, the number one, G1 checkpoint, which is also called as restriction or R point. What is this restriction or R point? It is a point in G1 phase at which the cell becomes committed to the cell cycle, and after which extracellular proliferation signals are no longer required. This is the characteristics of the R point or the restriction point mostly. The restriction point is present at the G1 phase, and it is the phase in which the cell becomes committed to the cell cycle, and after which extracellular proliferation signals are no longer required. Extracellular proliferation signals mean, you have studied signals transduction. There has to be some signal to be initiated within the cell first that can trigger the other responses. Cell signaling transduction ultimately give responses to the cell. So the response here will be maybe cell division, or maybe entry to the next point or next phase of the cell cycle. So for the cell cycle to continue or to further enter into the next phase of the cell cycle, some extra proliferation signals are to be there. Signal transduction maybe through signal transduction, but but at this restriction point or R point here, it is a point where the cell itself becomes committed to the cell cycle, and after it gets committed, it no longer proliferation signals are required. Okay, so that means at this point we can say the cell is totally self-sufficient, or the cell becomes sufficient for to enter into the next phase of the cycle. Okay, another one, while studying this cell cycle, you will come across some name of some specific genes or like that, that is important for the cell cycle restriction and control of the cell cycle. One such example is P53, and this P53 gene is called as the guardian of the genome. Why it is called the guardian of the genome or guardian of genome? Because it can impose cell cycle arrest at any point when it finds that cell is not sufficient, or cell is not prepared to enter into the second stage. And as it imposes cell cycle arrest, it can induce apoptosis through its ability to, number one, to induce the expression of cell cycle inhibitors to prevent proliferation of the cell until it until any damage has been repaired, or it can initiate apoptosis if there is any genomic damage, if the genomic damage is too great and cannot be repaired. Obviously, if there is a lot of genomic damage, or if some after replication of the DNA, some DNA is not good, or it's there is some damaged DNA, obviously that damaged DNA has to be repaired, and that repair is done by many other by many other processes or methods that you will be study on genetics, molecular genetics. That is another different part. But if we don't control that DNA damage, that the cell cannot be repaired, we can't send the cell to the next stage of the cell cycle. That DNA damage and DNA repair has to be done, or has to be prevented, for the cell to go into the next stage of cycle. If and these all are mostly controlled by P53 gene, which is called the guardian of genome. Okay. Then in short, I will be showing you the slide. Don't worry. In short, I am now just telling about what are cyclins and cyclin-dependent kinases. This cyclin and cyclin-dependent kinases are mostly, they are very catalytically active components of the cell cycling mechanism. These are mostly cell cycling dependent kinases. From the name, you can understand these are enzymes which are mostly the active components, and these CDK, in short form, it is known as CDK. These CDKs are mostly the active components of the cell cycling mechanism. These proteins, these cyclins plus CDKs, these total unit, that enzyme part and the protein part, the total making the total part, total making it as a total active component. This, or you can say as mostly these are proteins. These proteins, mostly what they do, these proteins regulate the activities of other proteins by transferring phosphate groups to them. Mostly phosphorylation activation activity of the CDKs depend on the presence of the cyclin. Means the phosphorylation activity of CDKs, they they are mostly dependent on the other part, that is the cyclin part. Okay, and this cyclin mostly enable CDKs, means cyclin dependent kinases, to carry out the function to form a by forming cyclin CDK complexes. And when these cyclins are absent, mostly these complexes cannot perform any function, means they are functionless. Uh, and CDKs are mostly inactive in that case. So we can see uh, for the whole reaction to occur, we need both cyclin plus CDKs. If one of the component is absent or one of the component is inactive, the whole protein part, the whole system will become inactive. Means they, cyclin and CDKs are dependent on each other for function. They can't function isolately, either as cyclin or either as CDK. They has to be performed in concert. It means there has to be, they has to make a group, and they then function properly. So progression so through the cell cycle, it requires mostly alternate formation and degradation of the cyclin CDK complex. So formation and degradation, that are a continuous process of the cyclin CDK complex, that are going through the ongoing cell cycle process to occur. So this is mostly about what you have to study, you about cell cycle and its regulation and its checkpoints. What is cell cycle? What are the different cell cycle phases? What are the times needed for the cell cycle phases? What are cell cycle checkpoints? And what are the molecules that are playing the key role for this cell cycle even to occur? Okay, these are the main things that you are going to study in this topic. Okay, let me share you the screen. Okay, is it visible? The screen is visible, the ppt is visible to you all?
Yes, ma'am.
Yes, ma'am.
Okay. Okay, then cell cycle and cell cycle checkpoints. So what do we what how can we define this? We know that an organism which is a multicellular in nature, it needs to coordinate cell division across different tissues and organs, and these cell division or this whole cell cycle process mostly, what does it do to the cell? It's critical for normal growth, development and maintenance. And this and under this uh, topic of critical for normal growth, development and maintenance, what coordination has to be done? The coordination or coordinate timing of the cell division has to be maintained for proper cell cycle to occur. Then coordination of the rates of cell divisions has to be there. Not all cells can have the cell cycle. This is very important. Not all cells inside our body have the same type of cell cycle, or maybe the timing of the cell cycle may be different for all the other different cells in the body of a multicellular organism. Then frequency of cell division. What do we mean by frequency of cell division? Frequency mean how, in what interval the cell division occurs, or the phases are occurring. That that is termed by frequency of cell division. So the frequency of cell division varies. We can see that in a multicellular organ, different cell or organs or tissues, anything you can say, that can divide by its own nature or by its own timing. So as for example, if we see in an embryo, the cell cycle, we will be seeing that total cell cycle will be less than 20 minutes. In skin cells, in case of skin cells, the skin cells divide frequently throughout our life. Obviously skin cells, we need very much skin cells, because the skin cells usually are becoming dead. So to replace that dead tissue or the dead skin cells, we need rapid division of the cells of the skin, and that division is very frequently. It's mostly 12 to 24 hours cycle. It is continuously occurring 12-24 hours cycle. Then liver cells also it liver cells liver our liver is performing different types of activities. And in order to continue that different type of activities, obviously we have to get a continuous supply of the liver cells. And for that reason, it has that property, or it has that characteristics to retain the ability to divide. But sometimes, it to keep it in reserve, we have to also keep some cells in reserve for some future activities, as mostly glycogen is stored in the liver cells. So for that also, some things has to be kept in reserve for the activities to continue. So liver cells mostly divide once the maybe we need that, but the division occurs once every year or two. Okay, in this way the liver cells divide. Mature nerve cells and muscle cells, we know that nerve cells do not divide at all after maturity. Mostly the nerve cells, it remains permanently in G0 phase. Mature nerve cells and muscle cells, and this is the mostly the very much interesting characteristics of the nerve cells. We know that nerve cells do not divide. That is one of the important things. That's why many regeneration of the nerve cells are going on. People are doing regenerating the nerve cells, because we know that nerve cells do not usually divide naturally. So if we see the cell cycle, mostly it is a cyclic structure, a cyclical phase of events, where we will see that G1 phase. And what is the G1 phase? It's a cell in G1 phase. What happens? It's mostly the cellular contents, excluding the chromosomes, are duplicate. Only the cellular contents are duplicated. No chromosomes, nothing. The DNA nuclear materials are dividing in this phase. Then we will see the ACE phase, which is a very important phase, which is called as synthetic or synthesis phase. So here what we will see, each of the, we know that we have 46 chromosomes in human body as autosomes. So as the whole total chromosome as 46, 44 plus 2, 44 is autosome, and 2 is the sex chromosome. That is the total division of 46. So each of these 46 chromosomes is duplicated by the cell during this ACE phase or synthetic phase. That is why we call it as synthesis phase. Chromosomes are duplicated. It is replicated, duplicated. That means the DNA, the nuclear material we are getting, the nuclear material for we are getting, or the DNA material is synthesized in this during this ACE phase. Then coming to that, we will get G2 phase. What happens in this G2 phase? Mostly the cell double checks the duplicated chromosomes for error, making any needed repairs. If there is if during ACE phase there is some that occurs, if there is some error that occurs within the chromosome or within the cell, then this G2 phase always double checks it, whether the DNA is all right or not, whether there is an error, or it is okay. So at this phase, it double checks the duplicated chromosome for error, and if there is some error, that error is needed to be repaired, and that occurs during this G2 phase. After that, after all this preparatory stage that occurs, G1 S and G2, then ultimately we will get mitosis. That is the ultimate cell division, where the division, ultimate complete division of the cell takes place. Then the cytokinesis process. Cytokinesis means the division of the cytoplasm, where the mother cell actually divides into two daughter cells. Okay, and after this occurs, then the cell will take some rest. The cell cycle arrest is there, means there is no growth, nothing is occurring in that phase, which is known as G0 phase. Okay. Okay. Then events occurring in the cell cycle, it is giving it. It shows here the description what is happening in G1 phase, S phase, G2 phase, and M phase, and the timings are also the most general timings of each phases are given in this slide. G1 phase, it's mostly growth and increase in cell mass occurs following cell division, devoted to metabolic activities associated with cell growth and preparation for DNA replication. It mostly lasts for 10 hours. Then in S phase, DNA replication occurs. Genetic material of each and every chromosome is replicated, and it mostly lasts for about 9 hours. Sorry. Then G2 phase, it's another growth phase, we can say. It's a post DNA replication phase, we can say, where there is repair to damaged DNA also occurs, and it lasts for 4 hours. Then ultimately the final M phase, it's the final part of the cell cycle where two sister chromatids separate from each other, one going to each of the two daughter cells. Two daughter cells are mostly identical, genetically identical to each other and to the parent cell, and this M phase takes only 1 hour to complete. Now we are coming to the cell cycle control. How cell cycle is controlled? What are the major things that are controlling the cell cycle? Here we will get two irreversible points in the cell cycle. One, the two irreversible points are where we get the replication of the genetic material. It is not reverse, it is irreversible. These major events are not reversible in process. These are irreversible, means if replication of genetic material occurs, it occurs, and it is the constant event that is occurring, most concrete and constant event. No irreversible occurs. So irreversible points are here. It's one is the replication of the genetic material, and another occurs during the M phase, that is in the separation of sister chromatids. Then coming to checkpoints. These checkpoints are processed at the points where the process is assessed, and if there is any error or anything wrong is there, it's mostly halted. It does the cell cycle checkpoints are the points where the cell is entered into some arrest point. Where the growth has to be, or the events has to be halted to make the cell capable of going into further further stage. Then in cell cycle checkpoint control system, if we see the checkpoint in the checkpoints, what we see, the cell cycles are controlled by stop and go chemical signals at some critical points. So the stop point may be given by a red signal, and a go signal, like the traffic signal, go signal can be given by green signal. Okay, and these are the signals where the signals indicate if key cellular processes have been completed correctly or not. So these are the checking points where we have to check whether all key cellular processes have been completed correctly or not, so that it can enter into the next stages of the cycle. So that's why we call it as a control system. So the major three checkpoints already told about this. One is happening at G1 slash S phase. In between one checkpoint is occurring in between the G1 and the S phase. Another one is occurring at G2 and M phase, and the final we can call, that is occurring in metaphase to anaphase stage, that we call as spindle checkpoint. So what is happening in this all three major checkpoints? G1 slash S phase, we are questioning to the cell, can the DNA synthesis begin now? Is the cell, sufficient, is the cell is self sufficient to go for the next cycle? That is the question we ask during the cell cycle check major checkpoints. During G2M phase, we also ask one question: has the DNA synthesis been completed correctly? Is it committed to mitosis? Is it going? Is it capable of going to the next stage of next stage of the cell cycle? That question we are asking during this major checkpoints. Then in spindle checkpoints, what question we ask usually: are all chromosome attached to the spindle? Are all chromosome, they are attached to both the spindles of that are forming, that are known as pole formation, that are forming during cell division? Are that okay? Can sister chromatids separate correctly so that the sister ultimately separation will occur? And can then sister chromatids separate that correctly? These are the questions we ask during the checkpoints. So G1 and S checkpoint is the most critical. It's mostly the primary decision point. That is why we call it as restriction point. And if the cell receives the go signal, then it will go and it will ultimately divide. What are the internal signals? Internal signals mean that there will be the cell growth, cell size. That means ultimately increase of size, number or number increase we will see. Cell nutrition, whether the cell is whether there is enough nutrition so that every processes can occur. And external factors, we all know these are growth factors. That is also required for all this cell cycle also. And if cell does not receive any signal, if it does not receive all these internal and external signals, then the cell can understand that I am not committed, I am not prepared, so I will enter into, I will exit the cycle, and I will switch to G0 phase, which is the cell cycle arrest phase. And the function of the Z0 phase is non-dividing, and it completely stays in rest, and it's in this during this G0 phase the cell stops dividing, and it remains as it is, not doing any function. Then mostly G0 phase, we call it as non-dividing, differentiated state, and most human cells remain in this, mostly human cells remain in the Z0 phase. As for example, liver cells that are divided in a long, in a very much greater interval. That's why the liver cell mostly remains in the G0 phase. It divides, the liver cells is usually divides once or twice in a year, or means once, very long term frequency is there, not in short frequency they divide. Nerve cells and muscle cells also mostly remains in the G0 phase, because we see that nerve cells and muscle cells do not usually grow. They are very highly specialized type of cells, nerve cells and muscle cells, and they mostly remain arrested in G0 and can never divide. Okay. Then coming to the activation of cell division. How do a cell in its division stage gets activated? So how do cells know in when to divide? The cells also has to know themselves by themselves that this is my time, the time has come now for me to divide. So how this occurs? It occurs by cell communication signals. Cell communication organelles you have already studied before, and there are maybe the signals are coming from that organelle. Cell communication, cell to cell junction, gap junction, nexus, add cell adherence, all this, the specialized structures, these give signals, and after this cell communication signal is there, they get some chemical signals in the cytoplasm which gives them cue that it is now the time you can divide. And signals usually mean everything, you know, is everything is proteinaceous in nature. These signals are also signals means proteins, and these proteins can be activators, means which activates the reaction or which further catalyzes the reaction to occur. And some signals can be inhibitors too, means they inhibit the reaction to occur. Okay, then go ahead signals, they are internal signals, we call that promoting factors which promotes the reaction. External signal, we call it as growth factors. And then the mechanism, and then there is also mechanism of control. And the primary mechanisms of control are mostly, we know, phosphorylation, that we have already seen. Phosphorylation is also one type of signal transduction, and for that what do we need? We need kinase enzymes, and these enzymes either activates or inactivates the cell signals. Then coming to the main enzymes or kinase enzymes, what are those? Those we will see as cyclins, CDKs, and both cyclin CDK complex. Okay, so cyclins are mostly regulatory proteins, and they level cycle in the cell. They will adjust the cycle within the cell. And CDKs are cyclin dependent kinases. What do? They mostly phosphorylate cellular proteins. Ultimately that inactivates or activates the proteins. Okay, and the total CDK cyclin complex, the total complex, it triggers passage through different stages of cell cycle. It helps the cycle to move to the further different stages of the cell cycle. Okay, so this is the picture of what are the different types of cyclin and CDKs that are present at which point of the cell cycle, and what does it helps, that is given in this slide. We know that in G1 after G1 phase, we will get some cyclin D, which is the regulatory protein, along with the enzyme CDK4/6. These are the names given to differentiate between different cyclin and CDKs. Okay, so cyclin D and CD4K and 6 that are activated after G1 phase. Then there is also another phase that is called cyclin A and CDK2, that also that is also present between G1 and S phase. Then between S and G2 phase, cyclin A or B along with CDK1, that is working within S and G2 phase. Then within G2 and M phase, cyclin A and B or cyclin cyclin L. It is given as question mark because it is still in research level. Research is going on whether it is whether this really this cyclin L is functional at this stage or not. Then there are cyclin C, CDKs, cyclin C, CDK2 that are occurring in the G0 phase, which is a go, which is operating before G1 phase. Okay. Then what are the cyclin and cyclin dependent kinases? The CDKs and cyclin mostly drive cell from one phase to the next phase in the cell cycle, and these are important for proper regulation of the of the cell cycle, so that the genes for these regulatory proteins has been highly conserved in nature through evolution. Means the reaction that are occurring, that has to be very specific, and they are to be very highly conserved. they are they are not to be mixed with other types. That is why it has to be highly conserved in nature. And the genes are basically the same what we get. It's basically same in yeast, insect, plants and animals, including humans. Okay, the genes that we are seeing, that are occurring or that are helping in our cell cycle process, all are mostly conserved. And they, as it is conserved, they are of same nature in yeast, insect, plants and animals. Then cell cycle checkpoint regulation. If we see what are the checkpoints, what are the factors that they are checking. Mostly G1 checkpoints, they check for the cell size, nutrients, growth factors, DNA damage. Then for G2 checkpoints, what they mostly check, they check for cell size and DNA replication. And for spin and for the spindle assembly checkpoint, that is occurring in the M phase mostly, during M metaphase to anaphase stage. What that checks for, that checks for chromosome attachment for spindle, for to spindle, whether the chromosome is rightly attached to the spindle fiber or not. So these are the checkpoints regulation, what actually they are checking for, what are the factors they are checking point, they are checking, the cell cycle checkpoints are checking for. Okay, so with this we come to the end of this topic as of cell cycle and cell cycle checkpoints. Okay, so it's all done for cell cycle. Tomorrow we will be doing the last topic, that is cancer. And that is the mostly the last topic of your cell biology. So
Ma'am,
any, হ্যাঁ বলো.
ma'am, what are the factors which can affect the timing of the cell cycle to increase and halt?
To increase and halt means মানে তুমি কি বলতে চাইছো, টাইমিং এর জন্য কোন ফ্যাক্টর গুলো নিডেড?
Yes, ma'am. Yes.
টাইমিং এর জন্য দেখো, একটা সেল যখন ডিভিশন শুরু করে, ওভারঅল টাইমিংটার উপর অ্যাজ সাচ হয়তো ফ্যাক্টরস আছে। যদি দেখতে চাও, মোস্টলি ওই নিউট্রিশনাল গ্রোথ ফ্যাক্টরস গুলো ইম্পর্টেন্ট। একটা সেল যদি ফুললি ম্যাচিওর ফর্মে না থাকে, সে তো তার মানে মানে আমি বলতে চাইছি যেটা, যে রিলেশন বিটুইন নিউট্রিশন and the timing or the frequency of division, একটা সেল যদি ফুললি ম্যাচিওর থাকে, ফুললি সাফিসিয়েন্ট থাকে, অবভিয়াসলি তার তার তোমার টাইমিংটা, ফ্রিকোয়েন্সি অফ সেল ডিভিশনটাও ঠিকঠাক হবে। মানে এভরিথিং আলটিমেটলি ডিপেন্ডস অন দ্যাট নিউট্রিশন, গ্রোথ ফ্যাক্টরস, কেমিক্যাল signal that are required, means those whole proteins that are present in the cell। মানে একটা সেল যদি ফুললি প্যাকেজড থাকে, প্যাকেজ কথাটা বলছি কারণ কি, সবকিছু যদি তার মধ্যে থাকে, তাহলে তার টাইমিংটাও ঠিকঠাক থাকবে। মানে যেগুলো স্পেসিফিক টাইমিং সব সব ফেজের জন্য দেয়া আছে, সেই ফেজ, সেই টাইম সবকিছুই যদি ঠিকঠাক থাকে, তাহলে সবকিছুই সেরকম ভাবেই মেনটেন হবে। মানে অর্গানাইজেশন গুলোও ঠিকঠাক করে হবে, সেটাই আমি বলতে চাইছি। মানে ওই তোমার কি বলে, নিউট্রিশন, তারপর গ্রোথ ফ্যাক্টর, সবকিছু মেটেরিয়ালস নিডেড ফর ডিভিশন, সবকিছু ঠিকঠাক থাকলেই সবকিছু আলটিমেটলি, তার মানে টাইমিংটাও আলটিমেটলি ওইগুলোর উপরে ডিপেন্ড করছে। এই আর কি। আর কিছু? ইজ ইট ক্লিয়ার? ইজ ইট
Yes,
ক্লিয়ার?
ma'am. Clear. Clear.
ওকে। আর কিছু, আরো কারো কিছু যদি জিজ্ঞাসা থাকে, ইউ ক্যান আস্ক।
ম্যাম বলছিলাম কি যে প্রিভিয়াস ইয়ারের যদি কোয়েশ্চেন পেপার কোনোভাবে
প্রিভিয়াস ইয়ারের কোয়েশ্চেন পেপার আমি অলরেডি আপলোড করে দিয়েছি। চেক ইউর গুগল ক্লাসরুম। অনেক আগে আমাকে ক্লাসে বলেছিল, আই ডোন্ট রিমেম্বার কে বলেছিল, আমার মনে আছে সেকেন্ড থার্ড ফোর্থ দিনেই আমি আপলোড করে দিয়েছি। একটা দেখবে এক্সেল শিটে আছে, সেটা হচ্ছে
আচ্ছা
লাস্ট ইয়ারের কোয়েশ্চেন। সেটা যেহেতু অনলাইনে সাবমিট করতে হয়, ওই জন্য আমাদের ওই ফরম্যাটে করতে হয়। ওখানে কোয়েশ্চেন উইথ আনসার্স, কারণ যেহেতু অনলাইন পরীক্ষা আমাদের আনসার গুলো প্রোভাইড করে দিতে হয়। তো
ম্যাম
সেটা
গুগল
দেওয়া,
ক্লাসরুমেই আছে তো?
হ্যাঁ হ্যাঁ, গুগল ক্লাসরুমের
হুম।
একদম প্রথম, মানে অনেক আগে একটু চেক করো, ওখানে আছে। আর তার আগের বছরে তখন অফলাইন পরীক্ষা হয়েছিল। অফলাইন পরীক্ষায় তো বড় সবই মিক্সচার অফ কোয়েশ্চেন পেপার্স হয়েছিল, অবজেক্টিভও ছিল, বড় কোয়েশ্চেনও ছিল, ছোট কোয়েশ্চেনও ছিল। তো সেটাও আপলোড করা আছে, সেটা দেখবে ওয়ার্ড ফরম্যাটে আছে। ঠিক আছে,
ঠিক আছে ম্যাম।
2 বছরের কোয়েশ্চেন দেওয়া আছে। তোমাদেরও অনলাইনই হবে, ওই অনলাইন পরীক্ষায় হবে। হোপ এভরি কোয়েশ্চেন উইল বি অফ এমসিকিউ ইন নেচার।
আর
তোমাদের
ম্যাম সেমিস্টার তো মার্চে হবে তাই না?
সেমিস্টার মার্চে, আমি যা কালকে মানে এই আগের সপ্তাহে জেনেছি যে মার্চে হবে, কারণ তোমাদের এখনো স্টিল অ্যাডমিশন ইজ গোয়িং। ইয়েস্টারডে ওয়াজ দ্য লাস্ট ডেট ফর অ্যাডমিশন, সেই জন্য তোমাদের বলে এত দেরিতে। কিন্তু তোমাদের ওই সিলেবাস তো শেষ হয়ে গেল। আমি জানি না যারা এর পর জয়েন করবে তাদের জন্য একটু মানে একটু প্রবলেম হবে, কিন্তু কিছু করার নেই। আমি তো আর বসে থাকতে পারি না তোমাদের, তো আই হ্যাভ টু কন্টিনিউ, এই আর কি।
ম্যাম
হ্যাঁ বলো।
বলছিলাম আমি কালকে নতুন জয়েন করেছি, তো আজকে আমি ফার্স্ট ক্লাস করছি সেল
ওকে,
বায়ো। যদি
আচ্ছা,
হ্যাঁ
কালকে
যদি কোনো
ক্লাস,
প্রবলেম হয়,
নিশ্চয়ই নিশ্চয়ই
কালকে
নিশ্চয়ই,
যদি
কিন্তু
হ্যাঁ যদি
কিন্তু
কোনো প্রবলেম হয় আপনার ছুটির পরে আপনাকে জিজ্ঞাসা করবো, তাই তো?
কি বললে আর একবার রিপিট করো, আমি কেটে গেল, ভয়েসটা
যদি
ব্রেক
কোনো
করলো।
প্রবলেম হয়ে যায়, হ্যাঁ শোনা যাচ্ছে?
হ্যাঁ হ্যাঁ হ্যাঁ,
যদি কোনো প্রবলেম হয় তাহলে ওটা আপনাকে আমি পরে জিজ্ঞাসা করে নিতে পারি
হ্যাঁ
তো?
পরে জিজ্ঞেস করো। আমি তোমাকে একটু ক্লিয়ার করে দিই। আমি আবার এই জিনিসগুলো, টপিকগুলো, যেহেতু তোমরা নতুন জয়েন করেছো, ওদের হয়ে গেছে এতটা, আমি জানুয়ারি মাস থেকে আবার ফ্রম দ্য ফার্স্ট ভেরি বিগিনিং অফ দ্য মানে টপিক আমি রিভিশন স্টার্ট করব। সেখানে ইউ উইল গেট দ্য চান্স টু, আই থিঙ্ক, টু ক্যাচ অল দিস টপিকস অ্যান্ড এভরিথিং। বাট আমার সবকিছু গুগল ক্লাসরুমে আপলোড করা আছে। ইন এনি কেস তুমি যদি কিছু নাও পাও, কোনো স্লাইড, আমাকে আমাকে জানিও, বা তোমাদের সিয়ার সৃজনী আর এখন জয়রাজ, ওদেরকে বলো, দে উইল প্রোভাইড ইউ, মানে ওরাও তোমাকে তুমি না পেলে তোমাকে যে স্লাইড গুলো তুমি পাওনি সেগুলো ওরা দিয়ে দিতে পারবে। ঠিক আছে, আর যদি কিছু না বুঝতে পারো টপিক, ডেফিনেটলি ইউ ক্যান আস্ক মি, বাট একটা কন্ডিশন, from next week to 26 December, please don't ask me. I am not able to satisfy your query because আমার নিজের পরীক্ষা আছে তখন, আমি এমবিএ করছি।
ওকে ম্যাম।
ঠিক আছে, তো ওই সময়টা আমার সেমিস্টার পরীক্ষা চলবে, তো তো আমি at that time I will be completely forget with cell biology and everything, because MBA তে আমার অনেক সাবজেক্ট, অন্য সাবজেক্ট আমাকে অন্য রকম পড়া, সেটাতে I will be absorbed in that. তো after that from 27 onwards you are always free to ask. ঠিক আছে,
হ্যালো ম্যাম।
ওকে
হ্যাঁ
ম্যাম।
বলো
তো
বলো।
ম্যাম বলছি সেল বায়োলজিতে যে গ্রুপ প্রজেক্টটা আছে
মানে
ওটার
পিপিটি,
সাবজেক্ট, হ্যাঁ
পিপিটি
পিপিটিটা,
তো,
ওটা সাবমিশন ডেটটা কবে আছে?
পিপিটিটা তোমরা জানুয়ারিতে দিও, কারণ যেহেতু আমি বলেছিলাম ডিসেম্বরের লাস্টে, বাট এখন দেখলাম তোমাদের যখন মার্চ মাসে পরীক্ষা, অনেকটা দেরি আছে, টাইম আছে। তো তোমরা আস্তে আস্তে তৈরি করো। মার্চ তোমাদের হয়ে গেলে, তোমাদের বলো when you are finished with that, তাহলে আমি জানুয়ারি মাসের মাঝামাঝি বা জানুয়ারি মাসের শেষের দিকে কোনো একটা টাইমে নিয়ে নেব পিপিটি প্রেজেন্টেশনটা। ওকে, আমি জানতাম না যে এত দেরিতে তোমাদের মানে সেমিস্টার, সেই জন্য তোমাদের বলেছিলাম ডিসেম্বরের শেষের দিকে, আফটার 26। বাট এখন যেহেতু টাইম আছে, ইউ টেক ইউর টাইম, ইউ ডু ইট স্লোলি, টেক ইউর টাইম. I will take it on at the maybe on the mid or the end of January. পিপিটি প্রেজেন্টেশন। ওকে,
তাহলে কোনো ফিক্সড ডেট নেই, মানে ওটা
না
টেন্টেটিভ আছে
ডেট
না?
নেই, না ডেট আপাতত দিচ্ছি না, কারণ কি যেহেতু পরীক্ষা শুনলাম এত দেরিতে, তো আস্তে আস্তেই তোমরা করো। ডেটটা আই উইল মানে আই উইল সেটেল ইট অন জানুয়ারি, জানুয়ারির প্রথম দিকে বলে দেবো ডেটটা। ঠিক আছে, ঠিক আছে।
হুম।
আর কি বলো, আর কিছু যদি জানার থাকে বলো। আর কিছু কি জিজ্ঞাসা আছে? তাহলে আই উইল বি সাইনিং অফ। ঠিক আছে, then tomorrow there will be the last class of cancer cell before the break, cancer. So tomorrow see you all at 10:30. Okay, then bye. Thank you all of you.
Thank you,
Bye.
ma'am.
Thank you.
Thank
Thank
you,
you,
ma'am.
ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.